Good evening, everyone. It's Dr. Sheftall again, or should I say good morning. It's after midnight. I had a tough day in surgery, but I wanted to do a presentation on the COVID-19 vaccine. And we're going to do it in the form of a question and answer session because I've received so many questions from friends. In particular, Gina from Alabama, who's a patron of mine. And so she wanted to know all about the vaccine and where I stood on it. So let's go ahead. Oops, I have not gone to full screen yet. I am sorry. I will get there in 2 seconds. I'm just still learning this thing. Okay, here we go. All right. This is the typical bona fides page. So you can take a look at it if you want, but let's go ahead. Of particular interest, though, is my Patreon page. So please take a look at that. Take a look at me on Patreon. I'd love for you to become a patron. All right. We've got about 10 or 12 questions to ask, and some of them are very, very important near the end. So just hang in there. We'll get to everything. Is it true that COVID's infection fatality rate is the same as the flu? Yes. They are both 0.1% or lower. And I looked up some statistics just to give you an idea of what that means. Is for a healthy person under 60, healthy, I mean no significant comorbidities, not obese, et cetera, the chance of dying in a single 9-mile trip in a car is greater than dying of COVID-19. For a child under 18 years old, but school age, so let's say above 5, between 5 and 18, the chance of dying from a lightning strike during your life is greater than dying from COVID at that age. Will more people die from the response to SARS-2 than from the virus itself? Again, SARS-2 and COVID are synonymous. And the answer is yes, by a factor of at least 150. And I'm going to very briefly give you an idea of what I'm talking about here and why that's the case. As of today, December 3rd, 1.49 million people have died of COVID worldwide. Due to the economic fallout from the lockdowns, not the virus, 140 million people will starve to death, according to the World Health Organization. So already that's 100 times the number that have died so far from COVID. 100 million new people will fall into the extreme poverty category, living on less than $1 a day, and that's from the World Bank. 650 million people are currently facing acute food shortages, and that's from the World Food Program. So you can imagine the problems here. 650,000 people just in the US are on chemotherapy at any given time, and 1/2 stopped this year. They stopped their treatment. 1/2 to 3/4 of all cancer screenings were not done while the country was in lockdown. More than half of US children did not go in for their vaccines this year. Of course, there were lots of deaths of despair. This includes suicide, opioid overdoses, alcohol-related deaths, etc. The powers that be suspended non-elective surgery. So things like, they discontinued elective surgery, not non-elective surgery. That's a mistake. I apologize for that. Things like breast lump biopsies, hip replacements, knee replacements, many things were not done this year during the lockdown. And so what happens is people get sicker and sicker because cancer surgery was not done, and they will die in the next few years because of it, tragically. Okay, remember when you think about the number of people who lost their health insurance, there are about 11 million people who still have not gotten their job back. They have no source of income. So things like maybe a breast lump or dysfunctional uterine bleeding that a woman may notice, she's much more likely to spend money on feeding her children than she is to go see a doctor for $200. And then if he thinks radiological tests need to be done, it gets into the thousands very quickly. So if you lose your health insurance, you start putting off things that you need to check. And of course, some of these things are malignant or have a malignant source, and those patients will not do well down the road. And that's another tragedy of this. So it's an enormous amount of morbidity and mortality that came out of the policies that we instituted to get us through this much, much more so than the virus itself. Dr. Fauci said 90% of the people in the US are still susceptible. Is that true? No. Dr. Fauci is basing that 90% number on the number of people who have shown a positive antibody test, okay? Many doctors have repeated this number, but it just displays kind of, and I don't understand this because of course, Dr. Fauci knows all the immunology, but for some reason, they're not accounting for T-cell mediated immunity. They're only looking at the patients who have tested positive for antibodies in their blood. And T-cell immunity is more important for a viral infection than B-cell immunity is. So I just don't understand when I see the TV doctors come on and say things like this. It just makes no sense at all according to the understanding of the human immune response that we have. So, you know, you can speculate about why that could be. And I really don't want to speculate too much. I wrote something out there, but it just doesn't make sense that they wouldn't recognize that many people are protected from T-cell immunity and cross-reactivity from other coronaviruses, which is all proven now. All right. Now, I did a little calculation to try to give people an idea of how many people are protected from the virus at this point in time. That was a very good question from someone. And I predicted that it was 155 million in the US about a month ago, when only 9 million people had been exposed at that time. Now it's about 11 million, I understand. So let me show you how I made the calculation, and then I'm going to tell you what that means. The WHO said that 10% of the world population has been exposed, okay? The world population is 7.8 billion, so that 10% of that is 780 million. If you divide 780 million by 45.7 million, which at the time was the number of people in the United States, according to Johns Hopkins, that had been exposed. I'm sorry, it's the number of people in the world, according to Johns Hopkins, that had been exposed. You get a ratio of 17.2. Okay, in other words, Johns Hopkins is undercounting by a factor of 17.2, according to the WHO. Then if you take the number that Johns Hopkins was advertising as the number of Americans, people from the United States that had been exposed, which was 9 million just a month ago or so. It's about 11 million now. And multiply it by 17.2, you get 155 million. And that's where that number came from. And I think the WHO is much closer to the truth than Johns Hopkins. They have undercounted the exposed people the entire time. All right. Now, where are all those people hiding? Well, I believe that at least 30 to 40% of Americans have immunity in place due to prior cross-reactivity with other coronaviruses, such as those that cause the common cold. There are 4 of them, and they're shown here kind of graphically. Now, let me give you some examples of what we both observed that kind of support this idea. Have you ever wondered why when a nursing home gets an outbreak and several of the people in the nursing home get infected and die, why everyone in the nursing home doesn't get it? It's a closed building. These people have full-fledged coronavirus, active coronavirus infections, and they're breathing it into the air. All of the nursing home patients, of course, are breathing it in. It's an anoscopic virus. It's going down the hallway on a waft of air. It's going in the cracks between the doors. Every time someone opens the door, the coronavirus goes in. So everybody's breathing it in. Why are less than 100% getting infected from this? And all the patients are old. They're not exercising very much. They have a lot of comorbidities. Why aren't they getting it? Well, I believe that 30 to 40% of those nursing home patients also have cross-reactivity, protection from cross-reactivity from common colds that they had gotten through the years. Now, you may say, well, do you have any proof? And the answer is yes. Let me just show you a graphic from a recent study that I took a look at. Now this is for, first let's look at the numbers for someone who got exposed to SARS-2. And they showed a 100%, where are my glasses? They showed a 100% T helper cell response. Okay, remember CD4 is the subset of cells that we call T helper, and they're different than T cytotoxic, which are the CD8 subset, the T killer cells. I'll talk a lot more about that tonight even. 70% of the people who were known infecteds had CD8 measurable response, okay? And this is for the spike proteins, which are the proteins on the surface of the virus that help it get into the cells. The M proteins here, which are the proteins, as I said in the immunology primer in my video entitled Stop the SARS-2 Stupidity, Please. And I wish everyone would go back and look at the immunology part of that. It's very important for tonight. The M proteins determine the radius of curvature of the capsule that the genetic material rests inside of the virus itself, okay? It determines the radius of curvature of the virus, lipid outside layer. The N proteins are proteins that stabilize the RNA inside the capsule, and the non-structural proteins have another job, very important, and they kind of hijack the endoplasmic reticulum. I'll leave that for there for now, but these are different proteins in the virus that the CD4 and the CD8 T cells respond to in everyone that's infected, 100% for the T helpers and 80% for the T killers, okay? And of course, the IgG response was present in 100% of those infected, and the IgA was present in 80%. IgA, remember, is the dimer that has a secretory component on it and allows the barrier protection. It's specific, but it helps the barrier protection because the secretory IgA is present in saliva and tears and so forth. Okay. Now, they also checked unexposed patients. These are patients who have never been exposed to SARS-2, and they've never been near anyone who's been exposed to SARS-2. 50% of those patients have antibodies in have T cells, active T cells in their blood for the spike protein, the non-structural proteins, and the M protein, okay, of SARS-2. And they've never been exposed to it, and they've never been with anyone who's been exposed to it. 20% of them have T killer cells for it, okay? So what's going on there? There's some kind of cross-reactivity going on with the other common cold viruses, okay? So I do believe that at least 30 to 40% of people have immunity in place due to prior cross-reactivity with other coronaviruses, which cause the common cold. Common colds are also caused by adenoviruses and rhinoviruses, but we're talking about coronaviruses here. And again, I pointed out that that's why not everyone in the nursing homes gets sick when several of the patients get sick. All right, now this is a great question too. What is an RNA vaccine and how does it work? So first, let me give you a little primer on what normally happens, what messenger RNA's job normally is, and so forth. And then you'll understand better how the vaccine works. Okay, so inside the nucleus, the DNA gets transcribed into RNA, okay? Then the RNA gets transported out into the cytoplasm. Okay, that's very important because a lot of people are concerned if they're injecting messenger RNA into me, could it possibly affect my DNA? Remember, the DNA is in the nucleus and the messenger RNA gets transcribed in the nucleus, but it comes out into the cytoplasm to interact with ribosomes. And let me talk about that now. Every 3 base pairs on RNA is called a codon, okay? And the RNA interacts with the ribosome in the following way. There's a moiety called transfer RNA that brings an amino acid into the ribosome and matches its anticodon with the codon on the messenger RNA. Oops, pardon me. It matches its anticodon with the codon on a messenger RNA, and it's when it comes in like that, it's bringing an amino acid, okay? There's a specific transfer RNA and anticodon for every amino acid that gets brought in sequence. And this is how the sequence of amino acids, which get connected together in this process, corresponds to the base pair sequence in messenger RNA. So the messenger RNA codes for a specific protein right here. And then the transfer RNA gets released to go pick up another amino acid if needed, based on the codons further downstream here, to put more amino acids onto the chain. Okay, and that chain ends up being the protein. All right, that process is called translation. So that's what normally happens and where it happens. Now let's talk about this vaccine for a second. The recipients of the vaccine are going to be injected with a messenger RNA, which is the red in this lipid coat right here. And I'll tell you why they do it in a lipid coat in just a second. But that lipid coat is going to deliver the messenger RNA into the cytoplasm of the cell. Remember, this is the nucleus down here. This is not happening in the nucleus. So I think that the risk that the messenger RNA that's getting injected into your arm, it's going to go into your deltoid muscle. The messenger RNA that gets injected is going to stay in the cytoplasm as messenger RNA. I'm just saying what should happen. There is an enzyme called reverse transcriptase, and it's possible that could switch the RNA back to DNA, and then something strange could happen. But I don't think that's a big problem. But maybe this is as good a time as any to say this. I'm going to be giving updates for this every week. And if there's anything else that I learn about this that I was either mistaken on or not sure of in the previous presentations, I will correct it and explain it. But it's supposed to stay in the cytoplasm and not affect your DNA. Now, let me continue on here. Since you know all about what happens, how we translate the messenger RNA into proteins, you can see that's exactly what happens here. Here's the messenger RNA that came from what was injected into your deltoid. We're now inside a cell. And by the way, let me quickly tell you why that happens. They put a lipid coat on this because the lipid coat can, for lack of a better term, meld into the lipid coat of the cell that all this is going on in and deliver the messenger RNA into the cytoplasm of the cell. If they just injected messenger RNA, it would have a hard time getting across the lipid layer because it's not fat soluble, okay? But the lipid layer is fat soluble, so it just sort of melds right into it and delivers the messenger RNA. But anyway, this messenger RNA is going to interact with the ribosome, just like it did here. The ribosome doesn't know, it just sees messenger RNA and starts tracking down, okay? And it's going to translate into proteins, just like it did here. But the people who designed the vaccine are going to make the messenger RNA correspond to the spike proteins on the virus. So this thing is going to, this little mechanism right here with the messenger RNA and the ribosome is going to make spike proteins, okay? Of course, it's going to design it so that it makes an antigen so that our immune system can react to the antigen of the virus. And they chose spike proteins. And there's a little bit of a problem with that, well, just let me tell you now. Spike proteins change more often than the proteins like the non-structural proteins or the N proteins because they're on the surface, and the virus is very good at mutating so that it escapes attack, if you will. And the spike proteins are known to mutate more than some of the other proteins of coronaviruses. So I'm not 100% sure why they chose spike proteins to be the ones at work here. So let me research that some more, why they chose the spike proteins. I see a problem with it immediately, and that's that they mutate a little more often. So what happens to the spike proteins? Some of them will go into a thing called a proteasome, which will chop them up into little peptides, okay? Peptides are fragments of proteins. Maybe they're 10 to 20 amino acids long. So those big proteins that make up the spikes on top of the coronavirus get broken up into little pieces in the proteasomes, and then they get presented in the context of the class 1 major histocompatibility complex protein. Now, this is really important, and let me tell you why. This is not stuck into the nucleus, by the way. This is just a crowded drawing. This is in the endoplasmic reticulum where this happens. But that major histocompatibility class 1 protein is going to be presented on the surface of the cell over here, and it is the MHC class that the CD8 T killer cells interact with. So this cell is going to be killed by that T killer cell in all likelihood, okay? It's going to go through a process called apoptosis and destroy itself. Now, I'm going to get back to a couple of reasons why that's bad in a little bit, but let me show you the mechanism a little bit more. That's a very important problem, though, issue. Now, some of the spike proteins are going to get released from the cell, evidently. I'm not sure of the mechanism of that, but they're going to get released into the interstitial fluid and then re-endocytosed back into the same cell, evidently, the way this drawing is. And then the protein that was released and then brought back in is being brought in from outside the cell. These were made inside the cell. These were phagocytized from the outside. And so these are going to be presented in the context of the class 2 MHC protein. Okay, as you can see here, they're going to be here. They're going to get broken into peptides. They're going to go onto an MHC class 2 molecule and be presented to T helper cells. T helper cells do not kill the cell. Okay, T cytotoxic cells do though, T killer cells. T helper cells, their job is to do basically 3 things. They release cytokines, which are important in many, many ways, and I'll get to that a little bit today, but I went to it in great detail in the other video, which I'd love you to go back and review. It's called Stop the SARS-2 Stupidity, Please. It's really important for you to review the immunology primer on that if you want to understand this a little bit better. I got to hurry today. But the T helper cells do 3 things. They release cytokines, which have a lot of activity. They stimulate the T killer cells, and they also stimulate B cells, okay, which I'm going to show you in the next slide. But they have to see the antigen. They have antigen receptors on their surface, and they have to see the antigen in the context of class 2. This is not unimportant detail that I'm telling you about here that you need to forget. You need to remember the difference between class 1 and class 2. Now, let's see if there's anything of use over here. Again, here's the messenger RNA in the delivery vehicle, which is a lipid membrane. Okay, here's the antigen. The antigen here is the same thing as the spike protein over there. It's just the generic term for it. It's the term that the receptors on these surfaces of the B cell, the CD4 T helper cell, and the CD8 T killer cell are going to recognize. Okay, remember this is all specific. This CD8 T cell here has many, many receptors on it, but they all react to the same antigen, the same epitope on the antigen. That's the same shape and kind of a lock and key type mechanism. In other words, they don't have different, they're called paratopes, but they don't have different shaped receptors on here. They all are looking for the same shaped antigen, okay? Same with the CD4 T helper cell. And in this particular case, the receptors on the CD4 cell are the same as on the CD8. So they're looking for the same antigen, of course, okay? They're looking for that spike protein shape. Again, the antigen gets processed through the proteasome to be presented in the context of class 1 to activate the T killer with the help of the T helper. And the T helper sees that antigen in the context of class 2, and it will go on to stimulate the T killer and stimulate the B cells, which we haven't even talked about yet, and release cytokines. So this is just a real quick summary of how the T helper interacts with the B cell. I'll go over it real fast because it's a slide I took from the other presentation. So you've got the antigen presenting cell, like I just showed you, showing the antigen in the context of class 2 to the T helper cell. Now that T helper cell is going to get activated when that happens, and it's going to go interact with that same antigen, remember, which has been recognized by the B cell. So the antibody on the surface of this B cell, at this point, it's acting as a receptor, not an antibody that's going to cause any problems. It's also an antigen presenting cell. So it presents the antigen in the context of class 2, and that stimulates the T helper cell to release interleukins, and they determine which immunoglobulin is produced by the various plasma cells. But they also stimulate the B cell to differentiate into those plasma cells, okay? And to form clonal expansion and memory. And that's the basis for vaccinating someone, okay? You're vaccinating them with something that stimulates them to clonally expand and be ready for the next infection by that same antigen, okay? Now, when the antigen that you've injected by way of messenger RNA going through translation to produce the antigen is a real foreign invader like a virus, there's another arm of the B cell stimulation called the effector arm. And that's when it goes and kills the invader, okay? Releases antibodies, and these antibodies go stick to it and it gums up the works, and other cells come along like natural killers and other things will come along and kill the virus, okay? So let me move on now. You got a good idea that the T helper cell is very important. Of course, the T cytotoxic is good and bad, right? There's gonna be a clonal expansion arm for memory for the next time the virus comes into the body, but it's also gonna kill some of the muscle cells that you injected the vaccine into, because those cells are presenting the antigen quickly in the context of class 1 right here, and that T killer cell is going to kill this muscle cell here, okay, which is where you injected that vaccine. So that's a little bit concerning, is it? And I'll tell you the biggest concern of mine at the very end. Okay, so let's continue on here. Someone asked, I had a positive antibody test. Do I need to get the vaccine? You should not need to get it, because you will have clonally expanded memory B cells waiting to attack the virus if it gets inside you again. And this memory lasts a long time. It's not the same amount of time that the antibodies last in your blood, because antibodies are just protein molecules and they have a half-life and they go away. But the cells that have clonal memory of that antigen that they were stimulated by initially, those last much longer. You'll also have T cell memory. Remember, the part of the immune system that's very important in viral infections, you'll also have that if you've been exposed to the virus enough to have a positive antibody test. So you've got a lot of protection already. And by the way, natural immunity is almost always much stronger than artificial immunity from a vaccine. For example, if you had the measles when you were a kid, you're not going to get the measles again. If you got a vaccine for the measles, you're probably not going to get the vaccine again. But the vaccine-mediated memory is not as strong as natural immunity, if you will, where you actually got sick from the disease and built the antibody response and the T cell response. So no, you should not have to get the vaccine if you have been exposed and had a positive antibody test. You shouldn't have to. Now, this is a real important slide. Great question. Is the vaccine safe for children and is it safe for adults? Well, I did a lot of research on this and I looked into the side effects that have occurred after the first dose and the second dose of 3 of the vaccines. And I was really disappointed. I was hoping these things were going to be real clean and there wouldn't be a lot of side effects, but especially for the second doses, the side effects were horrible. Horribly, let me say this, horribly common. I don't know how severe the side effects are. It doesn't really explain that, although they've interviewed a few people on the news who have described their difficulty with it. And it's very uncomfortable for about 2 days, and then they sort of feel better again. Now, 2 days would not be a huge price to pay. And if that's all that ever happens, that won't be horribly bad. But let me just list them and we'll talk about it a little bit. So for the Moderna vaccine, after the first dose, 10% had side effects after the first dose, but you need 2 doses for that vaccine. By the time they'd had 2 doses, every single trial participant was showing signs of headaches, chills, or fatigue. And for at least 80% of those people, those 100% that got side effects after the 2nd dose, 80% were ill enough to not to have it interfere with their daily activities. You know, they had to stay in bed, or they felt really bad, didn't want to go to work, and so forth. So that's pretty significant from a vaccine. We've all gotten vaccines before where you just feel a little sick the next day or two. Normally you go about your business. You know, you go to work, you just feel a little bit under the weather from a flu vaccine or something. And what you're feeling is that your body's immune response to the injection that you got. So in a way, it's good. You know, it shows you that your body's mounting a response, and it makes you a little sick when it does that. But, you know, we don't want it to be so bad that, you know, your fever gets really high and you get terribly sick from it, or get some kind of injury from it. For example, when I looked at the Pfizer data, a lot of the side effects were similar, but one patient had the chills so bad, he was shaking so much he cracked his teeth. So it's pretty serious, pretty severe. Again, if it lasts 48 hours and you can avoid something horrible like breaking your teeth, it may be worth it to you. And I'll talk about that later. And for the Oxford vaccine, 1/3 of the people that received the vaccine without acetaminophen is Tylenol, experienced moderate or severe chills, fatigue, headache, malaise, feverishness, and just over 1/4, just over 25% developed moderate or severe muscle aches. So all of the side effects seem to be fairly similar. Remember, these are the short-term side effects. There's no way we could know the long-term side effects of this stuff yet. And sometimes they're different and much more severe. Let's get on to that in just a second. Will I take the vaccine? All right, that was another question somebody asked me, which is a very good question to ask a doctor. If you want to know what medicines work, ask doctors what they're taking, because the doctors see so many patients that they get a very good sense of what works and what doesn't work. If you want to, it's just like really any profession. Okay, what kind of tires does a car mechanic put on his car? You know, that sort of thing. All right, so let's take a look at this for a second. My answer is not as it stands now. I saw a little bit too much that I wasn't too pleased with. I was a little concerned about a lot of it. And I'm going to give you the reasons in just a second. I might change my mind and do it later though. Okay, so here are the reasons. The short-term side effects were present in 100% of the trial participants after 2 doses of the Moderna vaccine. And that those side effects were severe enough in 80% of those people, that's 8 out of 10, to interfere with their daily activities. You know, it kept them home from work and so forth. Okay. The other thing that concerns me, and much more so than just being sick for 2 days, is that we don't know anything about the long-term side effects yet. And if you look into the history of vaccines, many vaccines were advanced and utilized with success, I will say. But if you look at the judgments that have been paid out by large pharmaceutical companies because of problems with vaccines, it'll scare you almost. It's shocking how many millions of dollars they've had to pay out for adverse long-term side effects. We've all heard about the issue of autism and so forth, and there's all sorts of mixed information on that, unfortunately, and it all needs to be looked into very carefully. I do want to say this, that's very important, and I'm glad I thought of that, because I neglected to mention it before when it was talking about, was it safe for children? I saw an interview with Dr. Fauci about 1 week ago, and somebody asked, and I knew this already because a good friend of mine from Fort Lauderdale, Lori, has 2 beautiful granddaughters, and she was asking me about the vaccine for her family and for her immediate family, her husband and herself and her son and daughter, and the grandchildren. And I told her, at this point, they haven't used any children in the trials yet. And children react to things completely differently than adults. They're different physiological machines, if you will. And just a week ago, someone asked Dr. Fauci if any children were in the trials. He said, no, but we're going to add them. And I certainly would not give the vaccine to a child, given the mortality rate, which is extremely low in children, say from age 5, not children, but up to 20 something. I mean, it's almost zero. Look at the data on that, please. But I think the CDC even has it at 99.997% survival for that age group. They haven't tested it on children yet. That would really scare me. I don't think I would give it to a child until I was absolutely convinced it was 100% safe. Given what I've learned about the vaccine industry and the awards or the judgments that have been paid out and all of this stuff really upset me. And I know there have been large vaccination movements to help the children in Africa, and there have been a lot of deaths from it. There's just no way I'd give it to a child yet. Okay. Because the risk of them having a problem if they get the disease is so low. I think it's lower than the vaccine risk. But anyway, let me continue on here. But the long-term side effects worry me much more than the headaches and the malaise and the fever and the muscle aches. We just don't know anything about it yet. It's too early. But here's something that upsets me a lot, or that makes me really worry and wonder. How do we know the vaccine gets injected into the arm where there's muscle cells? And those cells that I showed you where all the mechanisms were going on are muscle cells, okay? That's fine. And the major histocompatibility complex class 1 is going to present that antigen, okay, from the spike protein to the T killer cell. And it's going to see that antigen on the muscle cell, and its job is to kill that muscle cell because it knows that the invader, the virus, has hijacked the replication machinery of that cell, okay? And it's making millions and millions of viruses in there, and that T killer cell's job is to kill that cell. Okay, we're going to lose a little bit of muscle cells there, aren't we? We're also going to make clonal expansion to have CD8 T killer memory cells, which is a good thing. But here's what worries me about it. If you're getting a vaccine injected in your arm, what if some of the vaccine get into your circulatory system? You know, your veins or something, or your I guess small arteries could be in the muscles. Yeah, sure they can. And the person who delivers the vaccine gets some of the messenger RNA into your bloodstream, and it circulates to somewhere else in your body, and the same mechanism occurs, and the antigen gets presented in the context of class 1 to a T killer cell. Let's say, I'll be extreme just to make my point, but I don't know that this is possible. Let's say it's in your brain, and then the T killer cells is going to be killing some brain cells, okay? So, or maybe your heart valve, or well, not your heart valve, but your muscle cells in the heart, or your kidney cells or something. It could circulate around in your body, and the T killer cells don't know. If that cell presents an antigen in the context of class 1, the T killer cell is going to kill it. So that concerns me greatly. And that is if these messenger RNAs that they're injecting into your arm could get into your bloodstream and go somewhere else and cause death to other cells that are more important than muscle cells, which you can build back up, right? Okay, it's just an idea I had while I was going over this. And I thought to myself, what happens if the messenger RNA gets in your blood and goes somewhere where you don't want it to go? That scares me. I'd like to look into that, and hopefully I can present that to everyone within a week or so. I'd like to make an update of the virus issue every week so people can tune in and see about other things that I've discovered, and try to present it in a way that everyone can understand it. But that is the end of today's presentation. Thank you for watching, and please do visit my Patreon page and consider becoming a member so that I can continue to make this kind of good quality content and hopefully educate everyone in a fun and interesting way, and also an important way, so that you can better take care of your families when all this technology is zooming by us and we have to endorse it or not. And sometimes it's really hard to tell. I can imagine how much difficult it must be for a non-doctor to decide if they want to have this vaccine or not, based on all the incorrect information you're hearing every day from the media and stuff. So thank you again. And I'll see you down the road. Thank you so much. And please do look at the other video that I made and check me out on Patreon. Thank you.